Okay, in this podcast we're going to do a quick review of all the concepts that we learned in Unit 1, which was standards 1.1, 1.2, 1.3, 4.1, 4.7, and 4.8, basically the chemical basis of life. So what we're going to do is go over each of the essential questions that you are supposed to be responsible for. So number 1, you need to be able to identify the 6 elements that are essential to life by name and symbol. So remember, these are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So you got to know your chin-ups. Number 2, differentiate between atom, element, and molecular compound. That means how are these things related to each other? Easy way to say this: elements are made up of atoms, and molecular compounds are made of 2 or more elements bonded together. So that's how they are related to each other. Okay? 3, describe why carbon is essential to all life. Remember, carbon is the element of life. All living things are made up of macromolecules that contain carbon. Remember, the word that we use for that is organic. So all living things are made up of organic molecules. Those organic molecules contain carbon. Okay, so here we are with this question. What does organic mean? Okay, containing carbon. And organic molecules are what living things are made of. Now remember, you need to be able to identify all of the organic molecules by their basic structures. That is their monomer names, their polymers, and the major elements that they're made of. So this table can help you with that. So you have each of your groups of organic molecules here: carbohydrates, lipids, proteins, and nucleic acids. So the monomer of a carbohydrate is a monosaccharide. Many monosaccharides are a polysaccharide. The elements that make up carbohydrates are carbon, hydrogen, and oxygen, and some examples: sugar and starch. Lipids, the monomer of the fatty acid chains. Put the fatty acids chains together with a glycerol and you get a triglyceride. They're made mostly of carbon and hydrogen with a little bit of oxygen, and some examples are fat, oil, wax, steroids, and phospholipids. Proteins, the monomers of the amino acid. Put many proteins together, you get a polypeptide. You have the elements of carbon, hydrogen, oxygen, nitrogen, and sulfur. Sulfur is only in some of them. Enzymes and muscles are some examples. And then you have your nucleic acids. The monomer of a nucleic acid is a nucleotide. Put a lot of those together, you get a polynucleotide. Here you have carbon, hydrogen, nitrogen, oxygen, and phosphorus, and DNA and RNA are your major examples. Now, not only do you have to be able to identify the structure of each of these groups of organic molecules, you also need to be able to say what their functions are. So carbohydrates, sugar, starch, those are a source of energy. Cellulose makes up cell walls for plants. Lipids, fats and oils, energy source. Wax makes up your waterproof coverings. Steroids are various types of hormones, so you have your chemical messengers. Phospholipids we're going to find in our cell membranes. For proteins, a couple of examples. Enzymes are going to regulate our chemical reactions, mostly, remember, by speeding them up. Hemoglobin is an example that is going to transport oxygen, so lots of other proteins can transport stuff. There's also proteins called antibodies that fight diseases, lots of different proteins in your body. Nucleic acids, DNA, are storing your genetic information, and RNA is going to transmit that genetic information so you can do something with it. So those are some of the various functions for our organic molecules. All right, standard 1.3 was all about enzymes. Remember that enzymes are proteins. Okay, so the fact that enzymes are proteins means that they are made of amino acids, like all of our proteins. Okay, and the other thing about enzymes is that they fold into specific shapes, and those shapes are going to determine what those enzymes can do. So enzymes act as biological catalysts. To break that down, biological means living things. A catalyst is something that speeds up a reaction. So enzymes are going to speed up reactions in living things. Okay, so they regulate chemical reactions in living things, and they do that by lowering the activation energy of a reaction. So they make it so less energy is needed to start the reaction. Now remember, enzymes are also highly specific. One kind of enzyme for each kind of substrate, and substrate is the other name for reactant. The substrate is going to fit into the active site of the enzyme, and that's where the reaction occurs. So remember, we talked about that kind of like a lock and a key. There's one kind of key for each kind of lock. You can't open up any door with just any key. It's got to be specific. Enzymes work the same way. Oop, well, there you go. Enzymes also, remember, we're all going to work only under specific conditions. So all enzymes have an optimum temperature and pH. Remember, optimum means best. Enzymes in the human body tend to work best around 37 degrees Celsius. That's about 98.6 degrees Fahrenheit, and at a pH around 7, which is our neutral pH. So most of our enzymes in our body work best in these conditions. Okay. And remember, when we're talking about chemical reactions, the parts of the chemical reactions, the elements in a chemical reactions that are going to be rearranged are the reactants, and the reactants are going to become products. So reactants are going to become products, and that happens in every chemical reaction. Okay, and there's your reactants to products. All right, 4.1, we talked about the digestive system. And basically you have to describe the process of digestion and name the organs that are involved. This one's got a little bit more to it. So remember, digestion begins in the mouth, where we have both mechanical digestion, where we're physically breaking down food, and chemical digestion, where we're using enzymes like amylase, ends in ASE. The food then it moves down the pharynx and into the esophagus, down to the stomach. Okay? The stomach, remember, is mostly doing mechanical digestion by churning, and doing some chemical digestion with the enzymes and the acid, and we are forming chyme, which is your liquefied food at the end. From there, the chyme is going to move down into the small intestine, where lots of enzymes are going to be breaking down your food completely. The enzymes are going to come from the pancreas. You're also going to have some bile coming from the liver and gallbladder. And then the small intestine makes some of its own enzymes as well, and we are going to do the complete enzymatic breakdown of food, completely break it down. Now, this is also in the small intestine where our basic organic molecules are going to get absorbed into the blood and delivered out to each of our individual cells. So all our monosaccharides, our amino acids, our triglycerides, all of those things are going to get broken down completely and delivered out to our cells. And any undigested food and water is going to pass into the large intestine. The water will get reabsorbed into the blood, and then the solid waste will get eliminated out of the body through the rectum. Okay. Now recognize that macromolecules are converted into smaller molecules that can be used by the cells. These are your nutrients. So carbohydrates are going to get converted into our individual glucose molecules, and they are used for energy. Proteins are going to get converted into amino acids, and we can use proteins, these amino acids, for various things like growth and repair of the body. Our lipids, which mainly is fats when we break them in, we're going to break them down into their individual fatty acid chains, and we can use those to make cell membranes, steroids. They can store energy for us, and they also can provide insulation. So fats are an important nutrient in our diets. Okay. Now also we need to recognize the importance of enzymes in the proper functioning of the digestive system. Okay. Enzymes are used throughout the entire digestive system to increase the rate at which food is broken down. Without enzymes, it would take years to break down a meal. With enzymes, you can break down a meal within 24 hours. Okay. Now we produce enzymes at various places in the body. The mouth makes its own enzymes. The pancreas and the small intestine are going to make enzymes that are going to break down carbohydrates. Your stomach, pancreas, and small intestine are going to make enzymes that break down proteins. And then enzymes that are made in the pancreas are used in the small intestine along with the bile from the liver and gallbladder, and they're going to be used to break down fats. Okay. One last thing that we talked about in here was our homeostasis. So describe the electrochemical signals between nerve cells. Remember, we haven't talked about the nervous system much, but here's one neuron connected to the next one, and they don't actually touch. There's a space between them. That's called a synapse. And little chemicals called neurotransmitters are going to come out of that synapse, and that's going to allow one of these cells to communicate with the other. And remember, this process is very fast and it's very localized. So one cell communicates directly with the next one, and it's a very fast process. In contrast to that, hormones are chemical messengers that are made by the endocrine system. They have to travel in our blood, and they go to all the different parts of our body, and they're going to allow all the different parts of our body to communicate with each other. I have the word allow in here twice. Cross that out. There we go. Okay. Also in 4.7, identify that cells within tissues can communicate with each other without involving other systems. Remember, cells that are in the same tissue have chemical markers that allow them to recognize each other. So once a cell recognizes another one, similar cells can form gap junctions. These are like little bridges that allow them to send chemical messengers back and forth to each other, and that's how they communicate. And finally, in 4.8, explain the concept of homeostasis. So remember, homeostasis is maintenance of stable conditions inside the body. Okay, so things that we have to try to keep stable: temperature, pH, glucose levels, water. All of these things need to be kept at certain values, or you need to have a certain amount of them in order for your body to stay healthy. Also in this standard, we talked about the physiological feedback group. This is also called feedback inhibition. Okay, and most processes in the body are controlled by feedback inhibition. Okay, hormones, temperature regulation, these are all feedback loops. This means that whatever the result is of the process is going to stop that process, so it can't go on forever. So the perfect example of this: you are cold, you start to shiver. When you're warm again, you stop shivering. That's how it works. You don't just keep shivering forever. So the fact that the temperature changes is what causes you to shiver, and when your temperature changes again, you stop the shivering. Okay? So this is just a quick overview of all of the major concepts for Unit 1.